السلام عليكم ورحمة الله وبركاته. This is Dr. Omar Saeed. Inshallah, today I'm going to solve with you paper 6, 2025, variant 2. Can you please, do you hear my voice? If you hear my voice, just raise your hand, please. If you hear my voice, just raise your hand. Okay. Bismillah ar-Rahman ar-Rahim. Okay. We have talked before about paper 6 and how to handle paper 6. Today, inshallah, we are starting with May, June 2025, variant 2. As you remember, paper 6 is from 40 marks and it's 1 hour. Okay? So, always the first question is an experiment. He gives you an experiment and you start investigating the experiment and you start giving assumptions and you start giving answers for this experiment. Let's go for the first one and let's read it together. He's telling you a student investigated the effect of concentration of salt solution on osmosis. Once I see osmosis, I have to remember two things. Osmosis is the diffusion of water from an area of high water potential to an area of low water potential through partially permeable membrane. So we have three cases for potato. Whenever we have potato, we have three cases. The potato is to lose mass. The potato is to gain mass. The potato is stays the same. Okay? When does the potato lose mass? When the solution where it's added in is, yes, low water potential. So the water diffuses out of the potato. When does the potato gain mass? When the solution it's added in is high water potential. So the water diffuses into the potato. When does it stay the same? When it is nearly equal water potential with the solution. So water goes in and out with the same ratio. So let's continue. The student used three different concentration of salt solution. Whenever I go for the experiment, I have to look for one more thing. I, D, C, C. Please always remember this word. Whenever you go for experiment, you have to look for IDCC. I stands for independent variable. D stands for dependent variable. C stands for what was kept controlled or was kept the same. And the last C stands for your conclusion. So before I complete the questions, he's telling you a student investigated the effect of concentration of salt solution on osmosis in potato tissue. So what I am investigating, the effect of salt on osmosis. Let's say size of the potato, would it change or not? The mass, the length. So my independent variable here is the concentration of salt. My dependent variable that I'm looking for is the change, the size. Okay. So he had made a table. This is the table where he used more than one beaker. He used more than one beaker. These beakers were what? A beaker for the first one, which has S1, S2, S3, sample 1, sample 2, and sample 3. It may change the letters M1, M2, M3. يعني it has no relation with the answer. Okay, in the first one, it was the volume of 1.0 mole per decimeter cube of salt solution. Here, the volume of distilled water was 0, and here the final concentration of salt solution per mole per decimeter cube is 1. The first thing that he asks you to do is to get the concentration of salt solution and the volume of distilled water added to get the final concentration of the salt solution. Very easy. The volume of salt solution, it was 50. Final concentration was 1. So if the volume of 1 point 0 mole per decimeter cube of salt solution, it was 50, the concentration was 1. So the question comes like this. What if the final concentration of salt solution was 0 0.5? So what is the volume? Cross multiplication. Do you get it? Just the cross multiplication. When you compensate, it will be 1, okay? You just cross multiplicate and you get the 25, okay? But now we added 25 of the volume of the needed concentration. What is the volume of distilled water? The total volume that you need is 50 to make 50 centimeter cube. If you add it out of these 50, 25, the salt solution. So the rest is distilled water. So the rest is also 25. The concentration is now zero point 0.5. One more time. To get this, you have to compensate cross multiplication. Okay. Then after you get the cross multiplication, you get the salt concentration that you'll add. You will add only 25 of the salt solution. Then you will complete the rest by 25 volume of distilled water because the total volume that we need is 50. Okay. What if I need zero salt concentration? So I don't add any salt. I add only distilled water. So beaker 1, beaker 2, beaker 3, which is S1, S2, S3. S1, the concentration is 1.0. S2, the concentration is 0.5. S3, the concentration is 0.0. So this is the concentration that you gained. So next he said label the three beakers S1, S2, S3. Use the volumes of the solution S and distilled water W to make the table. You have done the table. Put the potato cylinders onto a white tile ceramic and cut each potato cylinder into 3 millimeter discs. When we have a potato like this, you will cut each 3 millimeter, you will cut a disc. Each three will cut disc. Each three will cut disc. And then you need 30 potato discs. Why? In this one, we will add 10 potato discs. S2, you will add 10 potato discs. S3, you will add 10 potato discs. After you add these discs, before you add them, you have to align all the 10 potatoes together and take the readings. Let's say that this one is 6.2. Place the 10 potato in a line next to a ruler as shown in the figure. Then take your measurement. Measure the total length of the 10 potato discs before you add them into the beaker. Number 6, put the potato in beaker S1, then repeat, which is cutting and taking the length, step number 4, and add the potato in S2 and S3. Then start the stop clock in the beaker of salt solution for 20 minutes. Leave them in the beaker of salt solution for 20 minutes. So all the potato were kept for 20 minutes. So we can consider that the time is a controlled variable. After 20 minutes, when you added the 10 discs of potato in each beaker for 20 minutes, you're going to do what? You're going to remove them, clean, uh, clean them or dry them, and line them along a ruler, the same like this step. So you measured before, then you will measure after. After you measure before and after, you can get the final total length of the 10 potato discs. Repeat for S2 and S3, the potatoes that were in S2 and the potato that were in S3. After you repeat, the student found that the total length was 0.102 millimeter. After 20 minutes, it was 94 millimeter. So the potato length decreased because the solution was what? 1.0, which is concentrated. So water You can tell me diffused out of the potato. Next, the S2 discs changed from 0.3 millimeter, 103 millimeter to 98. Also, the length of potato in beaker number two also decreased. So water also diffused out. Type. Last one, discs started with a total length of 100 millimeter. This was before submerging. So figure 1.4 shows the actual size of 10 potato S3 after the 20 minutes after submerging. You will get your ruler and you're going to measure this, and you have to give your measurement in millimeter. The measure will be 102 millimeter, which is nearly 10.2 10.2 centimeter. Okay, so you answered the first point. Next, prepare a table and record the result shown in the 1.3, this figure, and measurement, which is this measurement. Include the final concentration of the salt solution in your table. Okay, first of all, we have the length before, we have the length after, we have the concentration from here. That's it. You finished your table. When you design your table, the table will be like this. If you remember, we said, really, X-rays yield helpful, useful data. Okay, so you have to mention this part exactly. So you will write the salt concentration on the left side. Why on the left side? This was my independent variable. Okay. Then the final length on the right side. Why? Because this is my dependent variable. Okay. Next. Yes, the marking scheme gives a range for you. The marking scheme gives a range, but it's a range of plus or minus 1 millimeter. And if the answer is 102, the answer, the mark scheme gives you plus or minus 1 millimeter. So it may accept 101 or 103. But if you said 104, all of the answer will be wrong. Okay. Then you have the initial mass and the final mass in your answer. And if you want to get the difference, it will be no problem. Take care. You add the headings, you add the units, you add the correct data, and you add your measured. And if you I want to add here the beaker 1, S1, S2, S3, also it will be no problem. By this method, you ensured 3 of marks needed for this one. It's a mnemonic for the how to solve the table. I will share with you how to solve any table. Next, calculate the change in the total length. It's very easy. For S1, started with the 0.2, ended with 94. So it's what? 80102 minus 94 equals to 8. In Beaker 2, S2, started with 0103, ended with 98. So it's 5. This minus this. In Beaker 300, ended with 102. So it's minus 2. So state a conclusion. This was my fourth point I told you to get. Independent, dependent, controlled conclusion. What did you conclude from this? What did you conclude from this? What can you conclude from this? It's very important to conclude something. What is the thing that you can conclude from this? When we add what? Yes, who can tell me? When? In the market scheme, he said any one from these. What do you think? When the concentration was high, what happened to the length or the size or the mass of the potato? Yes, yes. The higher the concentration, the faster the osmosis occur, or the higher the salt concentration, the water is lost or moves out of the potato discs. This is one of the important points that you have to give, and that's why Cambridge is more convenient with the marking scheme. Gives you variety. Anyone from these. Also, you can say water moves into the potato disc when there is no salt, which is zero point 0.0, beaker 3, or impure water. Why it's pure water? Because no salt concentration, so it's distilled water. Okay? State the dependent variable. So as I told you, whenever you start the experiment, please solve these. While you go, underline them. So when it comes to the question, you already know them. It's a very unique word. IDCC. IDCC. What was the dependent variable here? Very easy. Yes, the length of discs. Mister, can I mention here size? No. Why? Because we measured the length. Can I mention volume? No. Can I mention mass? No. Because we measured the length in this experiment. Next question. Why the student used 10 potato discs? A very important tip I'm giving you in paper 6. Once he is asking why on a method or on a result, just give him directly reduce anomalous result. Just get it out from your pocket and give it to him to reduce the anomalous result. Once he tells you this question, why he repeated, why he used this instead of this, why he took an average, why he took a mean, just tell him to reduce the anomalous result. Cambridge said another answer. Any one from these. You may say that the difference in one disk is too small to be measured. You can mention this point also. Yeah, but if you mention this, you get your point. If you mention what I said, you get your point. If you say also one disk maybe not be representative, and so on. Describe one safety precaution. Whenever you see potato, so there is cutting, easy way to say cut away from your hand, habibi. This is what your mother always tells you. Cut away from your hand. All alternative wording. This, all the things that I write here is from Cambridge marking scheme. Then the student added few drops of yellow brown solution to test one of the potato discs. The solution became what? Blue black color. So we tested what on potato disc? Potato means what? Starch. What do we use with the starch? Iodine solution. What are the positive result? Positive result for iodine solution is blue black color. Type a negative result stays brown. Okay, so the solution is iodine solution. If you mention the word solution, no problem. It's between brackets. Conclusion: starch is present. Why? Because it said that there is blue black color. This means positive result. So this is question number one. Question C. A leaf is taken from a potato plant. Draw
Hello.
a larger diagram of the leaf. Sorry.
इसको बुलाएं.
Omar, you're
हाँ.
on. Next, draw a larger diagram of the leaf shown in this figure 1.5. Okay, whenever I draw, I have to go into something which is very important. Measurement. I have to measure. I have to measure. First of all, I use my ruler. I measure the length and I measure the width. This is the length and this is the width. Let's say, for example, and they must be this one. Let's say, for example, the length was 7 centimeter or 5 or 4, and the width was 5 centimeter. I will do the same thing. This point is very important. This technique is very important for you. to draw and get the correct proportion. Okay, but I will not draw this one as 7. I will draw it 9. I will add 2 centimeter or 1.5 centimeter. This one I will not draw it. Of course, this is not for scale because I don't have ruler here. This one it will not be 5. I will add also 2 centimeter. Let's say 7. Why do I do this? Because there is a mark from the 4 point marks that use the space provided more than 50% of the space provided. And if the leaf is like this, don't draw it like this, a small one. No, this is wrong. So this is the first point. The second point is what? When you have done this, try to remove the lines that you have drawn here with the eraser. But just leave the traces that you can follow. Then what? Then you have to know the correct proportional. Like what? This is the first quarter. This is the second quarter. This is the third quarter. This is the fourth quarter. So when you start drawing, you also are going to do the same. The first quarter, second quarter, the third quarter, the fourth quarter. Okay? So let's go for this. In this quarter, how many lateral veins are found? Three. So when you do the correct proportional and you start drawing, for example, you do the three veins like this. Don't do four. Don't do five. This is wrong. This is very important because some drawings, for example, he gives you a seed. I'm sorry, a fruit. Inside this fruit, there are seeds. For example, let's say that there are here 4 seeds, here 6 seeds, here 7 seeds, 8 seeds. So when you do this method, you will understand how many seeds are there, how many cells are there, how many layers are there, how many things are there. Okay, then you go for the details. What are the details? Take care from this in the marking scheme. Any main vein like this, you do it with double line. I need to remove this, but I don't know if you let me use a different color. Just a minute. طيب. So I have done my ratios. I have done my drawing. I have done my ratios. I start draw my leaf. I go by the parallel double line for the main vein like this. Then, as I know my ratios or I know my proportional, it should be there. I start my drawing. The line should be clear. Which line? The one that I'm drawing now should be what? Clear, not double line. And I don't draw the leaf like this with double lines. This is wrong. You're doing what like this? And you're using a pencil and you are not shading any point. I already shared with you the mnemonic. Who can please send for me the one that we told about it? Anyone from who attended yesterday's session? Send for me what we have said on drawing. What was our rule? Yeah, remember. Try to tell me. Then it must be what, yeah, mister? It must be a single line moving all over the leaf, and this single line cannot be broken. What does it mean by broken? Yeah, you don't do this. Yeah, and when you draw the leaf, don't do this. This is wrong. It should be one line single with no shading. One line single with no shading like this. And you have to know the proportional as I said. Yani when you measure this, it was 2 centimeter. Don't do it less than 2 centimeter and so on. So you start drawing the leaf, and by this you lose marks. Why? I'm sorry, but the eraser is not working. I don't know why. There are some details in the leaf that you have to know. What are the details? Yes. What are the details? The first detail is what? Yes. The first detail is these one, two, three, four. Three, four, like this, and one from the leaf is like this. So, why it's important, as I told you, to make these proportions? So, you know that these emerging parts of the leaf are what? Coming from proportional number 2 and 3. Not here, not here, and 2 here, and 2 here, not 3 here or 4 here. Okay? Then you do what? You do divination. Divination should be the same number of veins all over the leaf. Of course, this drawing is not, I'm trying to explain. I'm explaining, not drawing. So keep this in mind. So you start drawing the leaf as they are shown in the proportion. Then you do what? Then you make sure that the leaf side is what? Smooth. Don't do it like this. No, it's not jagged. It's a smooth leaf moving around. Okay, so go for parallel vein double line, single clear one using a pencil. Here, number of venation and the details of the parts that comes. So, Mister, what is the rule for drawing? Is there a rule? Yes, very important. Penguins do big, straight, labeled diagram. Exactly. Penguin do big, straight, labeled diagram. Exactly. Okay. BP stands for what? Use a pencil. Don't use a pen, even in graphs. Do, what's meant by do? Definite shape. Yeah, it's a clear line. Don't do any shading. Big stands for what? Must fit the space provided. S stands for what? Straight lines for labeling. This is a very important point. All of you, most of you lose marks. Now I want to label the midrib. This one. I am labeling like this with a arrowhead. Wrong. Nigis, we don't label with arrowhead. We label with just a line. Okay. So S stands for straight line, not arrowhead. L, label. This question is for 4 marks, so no labeling is needed, and he even didn't mention. But take care. Some past papers, the question turns with 5 marks. So there is 1 mark for labeling, and we use a line, not arrow, head. Then what do? We have the D, which stands for diagram. Must be double the size, double the size. Next, exactly count what? All the structures that is needed. Okay, is it clear till now? Okay, next, let's continue this. So also the tables use pencils. Line P and the Q represent the leaf in figure 1.5. Which leaf was mentioned? This leaf. There is a line here called P and Q. Use your ruler and you measure the line. The line, it is 63 millimeter, which is 6 centimeter or something like this, plus or minus. Okay. The actual length of the leaf is 39 millimeter. So he has given you the image size and he has given you the actual length. It's very easy. Direct compensation. What is the magnification? Magnification. What is the magnification? All the drawings in your paper, graphs, charts, whatever it is, using a pencil. Using a pencil. So, what is the magnification one? The magnification Very easy. Length of the line, which is image, over actual, I of M equal I over A. Direct compensation, so 63 over 39, because A is mentioned here, equals 1.6, and take care he needs it in two significant figure, so rounding up 1.6. 3 marks, 1 mark for the rule, 1 mark for the compensation, 1 mark for the result and the unit, but magnification has no unit. Figure 1.6 shows a leaf. This leaf is from a tomato plant. The tomato leaf is narrower than the potato leaf. I'm giving you now both leaves. Both leaves are here now. This is the one that we have done, which is the potato leaf, and this is tomato leaf. Compare the leaves. Describe one visible difference other than size. One visible similarity. Yeah. In the marking scheme, he said what? More veins on potato. Yes, very obvious. Fewer veins in tomato. Veins are more branched, veins are less branched. Here the edge is smooth. Here the edge is what? Jagged. Here, like this. Okay? Here it's one leaf, single leaf. Here it's formed from leaflets. Smaller leaves are from. Okay? Here is symmetric. What's meant by symmetric? You can get it into two identical halves. Here is not symmetric. The right half will be different than the left half. Okay? Next. One similarity. What is the similar between them? Both have mid-rib. Yes, you can say this. Both have petiole. Can you see the petiole? Okay. So also you got 2 marks from here. Any questions? Next. Enzyme pectinase is used in production of apple juice because it breaks down pectin in the cell wall. In an experiment, a study investigated the production of apple juice using 5 different concentrations of pectinase solution. Also, IDCC. Whenever I have experiment, I go for this. So we are investigating what? The enzyme pectinase and apple juice. So the independent variable, let's see. The in experiment, he investigated 5 different concentration. So we are controlling what thing? The concentration of pectinase. So this is my independent variable.
Teacher, your mic is muted.
I'm sorry. Can you hear me now?
Yes, we can hear you.
Okay. So now again one more time. How to control the temperature using thermostatically water bath. He measured the volume of the apple juice, which is the dependent variable. So my volume is the dependent variable. Temperature was maintained at 40 Celsius. Describe how the student could maintain the crushed apple and enzyme mixture at a constant temperature. As I told you yesterday, we have a list for, yes, the things that can be controlled, which is what? Temperature. Using what? Thermostatically controlled water bath. Any AVP, which means any valid point. Anything that the student can write and it gives the same idea, okay. State two other variables were kept constant. What was kept constant here? As I said, time was kept constant. What else was kept constant? What about apple? What about the pectinase enzyme? What do you think? Cambridge said what? The mass of the apple, which is 100 gram. Cambridge said what? The volume of bactinase or the enzyme. Yes, he added only 10 centimeter in the beaker. Okay, the time also was left constant. So if you say here the mass of the apple, it's correct. And if you say here the time, it's also is correct. The results are shown in this table. Percentage of concentration of pectinase and the volume of apple juice produced per centimeter cube. As I told you before, the left side is always what? The independent variable, which is drawn on x-axis. Thank you. And the right side is what? The dependent variable, which is drawn on the y-axis. Thank you. Okay, then we start drawing the table. Drawing the table is very important point that you understand. First of all, I need you to take a screenshot for the table. Take a screenshot, please, for the table. So we don't get back to it. Okay? So now you compare the results with me. Take a screenshot for the table. So now you compare the results with me. Okay? Yeah, let's go for this. After you have done taking the results, you should know that this will be on the x-axis, this will be on the y-axis. So the first thing that you do before you start drawing the graph, you do what? You write two things, the heading and the unit. Make sure that you write this on the x-axis and you write this on the y-axis. The heading and the unit. Yeah, let's do it together. The heading is what? Percentage concentration of pectinase solution. Percentage concentration of pectinase solution. Is there a unit? No, it's not mentioned. Here, the volume of apple juice and the unit is produced per centimeter cube. Produced per centimeter cube. By doing this and this, you got 1 point out of 4. Can you imagine? Number two, he needs a line graph. It's not histogram. It's not bar chart. Yesterday, we explained the difference. In histogram, they are touching each other. Same space. Same width. Is there spaces? No spaces. The bar chart, they are not touching each other. They must be the same width and the same spaces. Can you remember yesterday? Okay. Are both touching the y-axis? No. So, خلي بالي this we don't need. We need to understand the graph. Next, what is the next step? What is the second thing I do? I have to know the scale. What is the scale I'm going to work with? Anyone can volunteer and tell me what is the scale? How can we do the scale here? First of all, count the squares that you have. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4. 0 here. 1, 2, 3, 4. So, who can tell me the scale? How can we go for the scale here? For the percentage concentration, we have 1, 2, 3, 4, 5. We have 5, which is on the x-axis. And this one will be 1, 2, 3, 4, 5 also on the y-axis. Who can tell me? 40 on the x-axis and 60 on the y-axis. No, wrong. Someone said, just type for me in the chat. Ah, yes. Type for me. Type for me in the chat. Type for me in the chat. For the new people who have joined the meeting, I have sent in the chat the WhatsApp link where you can join and you can get the links for the upcoming past paper solving because we are solving for October, November. We are having revision sessions for October, November. الناس الجديدة اللي عملت جوين معانا أنا بعت في الشات لينك الواتساب جروب. في لينك الواتساب جروب ممكن حضرتك تلاقي لينك الواتساب اللي هننزل عليه ان شاء الله past papers solving past papers لاكتوبر نوفمبر. طيب so we have here four chances. My smallest is 0.1, my highest largest number is 1.6. How can we do it? So what do you think? طب I will give you a suggestion. 0.4, 0.8. Take care in the scale you have to be the same. يعني here you work it with 0.4 so it has to be the same. 1.2 here would be what? 1.6. Okay? The scale is up to the student, but there is one rule that you have to know, that your scale must cover more than 50% of the graph. This is also a point. So if you cover more than, you got the 2 mark. Of course, if this graph is right. For example, this is a graph and this is a graph. A student drew the graph here. Student covered more than 50%. This one doesn't get the mark, this one gets the mark. Okay, but let's go for this. Apple juice produced 4, 6, 12, 22, 22. How can we do it? We have only 1, 2, 3, 4, 5, 6. Because of the iPad, so the squares are not shown properly. Okay, yes, the scale is up to you. You can figure out the method that you want. Okay, يعني there is no in the mark scheme scaling point. No, it's up to you. The key is that it passes more than 50% of the graph and you are plotting the points correctly. So I will do it like 5, 10, 15, 20, 25, 30. Then I'm going to plot. As you remember, when I plot, I have two methods to plot. I plot with an X or I plot with a dot, then circle. Both are correct. Both very important. Please don't plot with a dot. Why? Confusing for the examiner, the one who is taking and checking your exam. Let's go for this. So, first point, this is 0.2, right? First point is 0.1 with 4. Let's go for 0.1. 0.1 will nearly be here with 4. Here we have 5 value and 10 square. So each square is what? 2. 1, 2, 3, 4. This is 4 and this is 0.1 nearly, right? So we go like this and we make our point like this. Then we have 0.2. 0.2 with what? Huh? With 6, right? So this is 5. This will be 6. We go 0.2 and 6. We plot. I think the point was 0.4 with 12. Yeah, let's go for 0.4. Up, 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 up. Each one with 2 points. So 12 is here. Here. Don't do these lines. Just doing these lines for you to clarify. Okay? Next. 0.8 with 22. Let's go for 0.8. Up, 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 up. Here with 22. And I think 1.6 was 22, right? The last point, yes. 1.6 with 22. This one with the same point. Okay. So now I have plotted my marks. I have plotted my degrees. I have plotted my things. Okay. Next, what can we do? We can now join the points. Please, please, when you join the points, use a ruler. Don't join the points with your hand. 1, 2, 3, 4, 5. What I have done is using my hand. This is wrong. I don't have a ruler. But use a ruler in your exam. Okay? Next. This will be the graph. I have to go out. Okay, just a minute. Use your graph to estimate the volume of apple juice produced when 0.5% pectinase solution. 0.5. Where is 0.5? This is 0.8, right? 0.4. Let's say that 0.5 is nearly 0.5, 0. Yeah, 0.5, 0.6, 0.7, 0.8. So 0.5 is nearly in this point. How do you do this? You go up, 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 up until you reach the graph or the graphing point. Then you go left, you will get the percentage from here. Of course, when you are doing it with the paper, it will be very accurate and you can get out the point. Type. So the graph should look like this. The graph should look like this. Okay? We will practice more graphs, inshallah. So here it gives you, look, because someone asked about the range. The range is from 13 to 15. Yani if you said 13, if you said 14, if you said 15, it will be accepted. I think that we got it 14 in the last slide. Depending on the candidate graph. So the graph is yours to do. Who can tell me what was the key that we used for drawing tables, for drawing graphs? We said that we have a key. Yalla, please send it to me. The people who attended yesterday's session, please send it to me. Yeah, who can remind me? People, describe the effect of pectinase on the apple juice production. What do you think? When the concentration increase, what happens? The volume of the volume of apple juice production increase until a certain point, right? Until a certain point. This point you can get it from the table. 0.8 nearly, right? So at this point, what happened? The effect of juice become what? Stop, increase, becomes constant. So it was increasing, increasing concentration of pectinase. You can write this, increasing concentration of pectinase enzyme. What did it do? Increased the production of the juice. Then became what? Constant or stop to increase. Okay? So whenever we draw a graph, we have to follow this rule. Okay? Yeah. What is the rule says? In dark landscape, smart printers find treasures. I D L S P F. I stands for independent on the X, dependent on the Y, label the axis, scale is correct, plotting using dots or X, fit the graph more than 50%, okay? And T for the trend, which is understanding what's happening in the graph. Easy? Pectinase is used to produce apple juice. So just what extra data will be required to determine if 0.8 pectinase solution is the optimal concentration? This question is always repeated. He tells you why 30 Celsius is the optimum, why 0.8 concentration is the optimum, why pH 4 is the optimum. Yani in this question, when he always asks you about a certain quotation or a certain date in the graph and he's telling you why we need to investigate more about this or why it is the optimum or why we have to make more experiment regarding this ratio, you just answer by a very simple question. You get the A star and the full mark. Use smaller intervals. Thank you. If it's telling you 30 Celsius is the optimum temperature, we need to make more investigation. Why we need to make more investigation? Or suggest what do we need to make? You tell him make small intervals above and below 30 Celsius. Make small intervals above and below zero point 0.8 concentration, zero point 0.7 concentration. Make small and so on. It's just standardized question with a standardized answer. Draw a label the apparatus the student could use to filter and measure the volume of apple juice. Very easy. You do this. What is this called in chemistry? You all know, huh? Who can remind with me? What is this called? Very easy. Yes, it's a flask. Thank you. Okay. Curve line on a graph. How can we draw a curve line? Okay, I'll solve this and I come to your question. Okay. So you say that this is the conical flask or flask. Then you drew the funnel. You call this funnel. Then you add something called filter paper and you make it with dots like this to separate it from the funnel. Filter paper. Then you add here the juice and here you say that this is the filtrate is filter. The mark scheme said that you also can draw the graduated cylinder. Draw and label. Why? So here, 3 marks. Okay? Next. The question that most of the students are confused with. Investigation question. We have something that helps us to solve any investigation question. Please copy it with me. Turtles fight snakes for many super incredible rewards. Turtles fight snake for many super incredible rewards. Very easy. T stands for trials. Prepare 3 test tubes. Prepare 3 spacement. Prepare 3 flasks. Prepare 3 beakers. So trial. What's meant by trial? More than one trial. F stands for independent factor. S stands for same factor. What was kept controlled? What was kept controlled? F stands for the second factor. We said independent. So the second is dependent factor. M stands for the method that you are going to measure the dependent factor with. S stands for safety. What's your safety precaution? I stands for improvement. How to improve? R stands for repeat. 1, 2, 3, 4, 5, 6, 7, 8. 8 points. In the question, he needs 6. If you mention 6 of these points at least, you will get the full mark. So I'm giving you 8. Okay. How to answer this question? Okay. Hydrogen, let me eliminate this part. Many super incredible rewards. Yeah, the first one trials. Hydrogen carbonate indicator can be used to determine the concentration of carbon dioxide in a solution. If the carbon dioxide concentration is high, the color is yellow. If it is medium, the color is red. If it is low, the color is purple. The table shows the color of the indicator at different concentration of carbon dioxide concentration. Aquatic plant photosynthesis and use up carbon dioxide when the light there. Aquatic plant respires to produce carbon dioxide in the light and in the dark. We don't need all of these. We just need this one. Use an aquatic plant to plan an investigation to determine the light intensity at which rate of photosynthesis is equal to the rate of respiration. So we will use what? What am I going to investigate? I'm going to investigate what? I will investigate color. How do I know? Because he said what? Carbon dioxide is produced and it's measured by hydrogen carbonate indicator. So I will use the color mentioned here as my dependent variable or dependent factor, and the light intensity is my independent variable. Very easy. But the first step is to identify who is dependent and who is independent. So he's telling me that there is a plant. I need you to make investigation for the light intensity. It's very easy and very common. Look, we get a plant, we add it in water, we get a lamp, and we add the lamp in different distance. Why? To control the light intensity. For example, for distance of 10 centimeters, 20 centimeters, 30 centimeters, and I move a lamp from one side or from one distance to the other. Once I find that the color of the hydrogen carbonate indicator inside the water changed or it became what? Red. Because when the carbon dioxide is equal to the oxygen produced, it gives me red color. Okay. Then I will stop the experiment, and now I know that carbon dioxide is equals to oxygen. So let's go for this. Let's answer this together. How to get it? Okay. We said that we go into these steps. Turtles fight snake for many incredible super rewards. So the first step is what? Writing the trials. Set up 3 test tubes. Set up 4 test tubes. Set up 5 test tubes. Each test tube contains or beaker aquatic plant with hydrogen carbonate indicator. You can mention the volume of the hydrogen carbonate indicator with 5 centimeter cube of hydrogen carbonate indicator. No problem. So you got 1 point. Number two, factor changed, which is my independent factor. So you will say that the independent factor is this in this experiment is to use 3 different light intensity. How? By placing a lamp at different distance, 10, 20, 30, from each test tube respectively. So you mentioned the second point. We mentioned the first, we mentioned the second. Then you will mention the method. How are you going to measure? How are you going to make it? Place heat shield. What's heat shield? When we have a lamp and we have a plant, the light will give heat, not only light. So we easily add beaker has water inside. So the water can absorb the heat coming and can reflect or refract the light intensity. So place heat shield between the lamp and test tube, prevent heating. Keep plants submerged by weighing it down for 10 minutes at each light intensity. So what was kept constant? The dependent factor. Observe the color of the hydrogen carbonate indicator in each test tube. The same variable or the controlled variable, same plant species must be used, same volume of indicator must be used, same temperature must be used for all the test tubes. Improvement. We use a colorimeter. What is this? Some people cannot identify the colors between yellow, red, purple, or the degrees between them. So we use something called colorimeter. Safety. Use safety equipments because here we have electrical equipment like light lamp. Secure the lamp stand. R. Repeat with 3 fresh tested tubes for reliability. Okay? And find the distance needed where the color stays red, so photosynthesis equals to respiration rate. So one more time, how to understand any investigation question. Any investigation question, I go for a technique called what? A technique called huh, turtles fight snakes for many super incredible rewards. I tell him, prepare 3 test tubes. I will tell you investigation for the effect of temperature on enzyme activity. A very common question. So, what am I controlling? Temperature. What am I investigating? The dependent factor, which is enzyme activity. If it's, for example, in yeast or cell respiration in yeast, so what will be produced? Carbon dioxide. So the volume of carbon dioxide produced is my dependent factor. Prepare 3 test tubes with different temperature. 30, 40, 50 Celsius. We answered the first point. The independent factor investigation. Just answering as am I a student in this investigation is the temperature where it will be controlled with different temperature 30, 40, 50. What should be kept the same? Controlled variables are I should control the pH for the enzyme. I should control the yeast species, yeast kind or yeast type species. I should control خلاص we are doing temperature, so I should control substrate concentration. I should control enzyme concentration. So all of these are controlled variables. Number 4, we finish this and this and this. The dependent variable in this investigation is volume of carbon dioxide produced as the effect of cell respiration of yeast respiration. Number 5, the method. How are you going to make the experiment? In the 3 test tubes, which test tubes mentioned above? Add, for example, a 5 centimeter cube of yeast solution. Use thermostatically water bath to adjust what? Different temperatures. What are the different temperature? 30, 40, 50 that were mentioned before. Mention the units for each. After this, use what? How are you going to measure the volume? Use gas syringe, right? Okay, to measure the volume of carbon dioxide produced. You get the idea? Number 6. So now we will understand how to get the method. Okay. Number 6, safety precaution. Is there any safety precaution? Yes, wear gloves. You always do it. Write this. Wear googles. Lab coat. Number 7. Improvement to maintain temperature. What do we use? Use thermostatically water bath. Okay? Number 8. يبقى we mentioned this, we mentioned this. Repeat. Take average. Repeat how many times? You have to mention how many times. 3 times at least and take average. Plot your data on a graph. And get the mean if you want to add. Get the mean. That's it. And this is how you solve any investigation question. If you follow these points, you will get the full mark. Inshallah. So this is for today. Inshallah, we will have another session. I will send in the WhatsApp group and we will solve some past paper, also paper 6, because your exam is coming for this point, Bismillah. Any questions? If you have any questions, I'm just